The name of God, the most gracious, the most merciful. Welcome. Today, we're talking about genes. We read about genes every day in magazines and hear about them on the radio. What's the story with genes? Simply put, they found that the secrets of our body's structure in every detail are written in our DNA. DNA is like a book of 5 million pages written in code. The complete blueprint for the entire body in the form of detailed instructions for building every cell, protein, and a hormone. But these instructions are called genes. The human genome map contains about 100,000 genes. Each gene is responsible for building a part of the embryo. Any error in a single code letter in this record immediately appears as a birth defect, a genetic disease, or cancer that suddenly appears in the colon, lung, or cervix. It turns out that there are also cancer genes in every human being. Everyone has genes, but these genes remain dormant, inactive, and latent, have no effect. What? Suddenly wakes them up and activates. Bad habits like smoking and alcohol addiction, or due to food contamination or pesticides or exposure to radiation. Suddenly, these genes wake up and cause cancer, resulting in the onset of a terminal illness. The idea now is to treat these diseases by introducing healthy replacement genes. How will they introduce the genes into the body? They put them in a virus and infect the body with this virus. So the gene enters the body. The whole world is now busy researching genes and DNA. Every country is trying to create a team to map the entire human genome via. The strange thing is that the word gene in Arabic is 2 letters. It remains kun, the divine command. And DNA is a miniature of the preserved tablet or hindi stored inside our cell's nucleus, containing all matters of destiny in life's journey. Tonight we'll explore DNA and genes, unveiling some of these amazing secrets, amazing holy secrets. Let's watch the film together. This boy is lucky because it was predicted he'd get retinal blastoma or retinal cancer. As the vial carries the cancerous gene. His father passed it on. So early signs were expected. They had him checked often and caught it early, starting treatments promptly. He's being sedated to examine his eye and see the radiation's effect. The cancer was in its early stages treated with deep radiation. They say the cancer is shrinking دائما. Nurses say he's lucky it was expected, and his family checked him often. He's sedated, and they'll examine his eye. Notice inside the pupil, that egg-like thing, that's what's left of the retinoblastoma, a quarter of its size, because we caught and treated it early. The family has a grandparent, aunt, or uncle who lost an eye to retinoblastoma. The gene runs in the family, passed down through generations. This is his father also carrying the gene, but he escaped it. A cancer gene doesn't guarantee cancer. It can be dormant, inactive, and do nothing. And it could lead to cancer. This is the father who discovered the gene in him. And this uncle had his left eye removed and replaced with a glass eye. And this one also had an eye removed and replaced with a glass eye. And this is the mother of the 6-year-old, and this is his father when he was young. The parents have great hope that the boy can overcome this and that the eye can be saved. That, instead of removing it, the cancer might dissolve and we won't need to remove it. As we said, the gene carries formative commands. So the cancerous gene carries a formative command for what? For cancer. But we said it might be dormant, complete, and inactive. And it might suddenly become active. It's clear that what? Vision hasn't been destroyed in the affected eye. The affected eye is the right eye, I think. And the boy can see the genes. We said that the genetic code is like a book of 5 million pages. In it is recorded all the secrets of the body's construction plan in detail. Every cell, every protein, every hormone, in detail. Look, he's taking a page from this amazing book. What do we find? It's made up of these code letters. These code letters are the formative commands for the body's plan. A mistake happened here. A C appeared instead of a T. This simple mistake is what caused the gene to turn into a cancerous gene, giving the command for what? For the formation of retinoblastoma. And he says, as an example, any finger that gets cut and bleeds, there's a gene called a control gene. It commands the fibrous cells to work and close the wound immediately. So you see the fibrous cells working, and they'll close the wound. Without that, one would bleed to death. So there's a gene behind everything. There's a gene behind all the processes that take place in the body. Every process has a gene controlling. So the whole world is now focused on discovering the plan. The genome. The human genome, which is the genetic code and its contents. And what are genes? And what is the function of each gene? It's called the human genome. And as we said, there are 100,000 genes. And each gene is responsible for a part of the embryo and a stage in its formation. And any error in these code letters means that a disease will appear at a certain time. Cancer or a birth defect or a genetic disease. Imagine 5 million pages filled with letters like this. See the plan here: to be a gazelle, a bird, a human, a butterfly, or an insect. The genetic code carries the whole plan. This is called Bob Weinberg. He was the first to discover the relationship between genes and cancer. And it's a really important discovery. And he did a dangerous experiment. The experiment he did was that he took the genetic code and then took the genes of cancer from a mouse with cancer. This mouse is sick with cancer, a cancerous tumor in the brain. So what? He took the tumor itself, and then he extracted from it the genes of the genetic code. Here, we'll see how he extracts the thread. This thread is the genetic code, and he put it in healthy mouse cells. And the result was very strange, that this genetic code gave an order to the healthy cells, so they all turned into cancerous cells. And this is strange. It really means that the gene carries an order and information. And this information, the gene has the ability to execute it. So it can turn a healthy cell into a cancerous cell. And here on the right and on the left, there are two strands, and we'll see one of them, which is the right one. There is a structural error or a code letter that came down wrong. Did it wrong. Any error, we said, in these simple letters means a different command immediately. We'll see here. Here, instead of T, A came. This A is what led to cancer. What happened in the mouse's brain? So when a piece of this gene is placed into normal cells, it immediately commands them to turn cancerous. This is the genetic code. And this code during cell division splits into two identical halves. And each half, watch how it splits. It splits like this in two. And each half regenerates itself. So we have two new codes needed for the two new cells because the cell divided. Thus, the genetic code renews itself with each division, making an exact copy. Secondly, they discovered receptors on the cell surface. These receptors receive growth commands, and based on these commands, they divide. Any defect in the receptors, see here how it shows a defect. A defect occurred in the receptors due to the gene. So, the receptors give spontaneous random division commands, many times more than usual, causing cancer. What affected these receptors? What made them deviate like this? The developmental gene. This gene, a cock party for Dr. Suleiman, who discovered the gene responsible for breast cancer. Women celebrated him greatly because he became their hope for treating cancers genetically or in a new way. So they held a party to raise donations for his research. He's the tall one standing, Dr. Suleiman, who discovered the gene responsible for breast cancer. American society is active and sensitive, supporting any glimmer of hope in any scientist, raising donations immediately. It's not government funds allocated here. No, the people themselves contribute. How many millions did they raise at that party? How many millions of dollars did they raise for him to continue his research? He says that now there's hope. We can beat cancer without radiation or harmful chemo. We want to first learn how cancer forms, what exactly happens, because if we know what happens, we can stop the process and prevent grand cancer right in Kupi. So we need to research and see, and he is. What caught his eye was that some cancers respond to treatment and some are very aggressive and don't. Some spread fast and some don't. So he said, let's see the difference in cells between mild cancers and stubborn, incurable ones. These are pieces from these cancers. Breast cancer. He grinds the cells to see the difference between resistant and responsive cells. First, he noticed the gene of the aggressive cancer cell. It leaves a strong, dark mark. See its shape here? It's called magnification or amplification. The gene gets amplified. Why? The gene's active copying itself, making copies. So the effect of this gene is doubled and amplified in molecular chemistry, cell sign chemistry. The gene leaves a shadow like this. See how the gene prints itself? It's copying itself quickly, so its effect is doubled. He said this gene makes these receptors. The patient whose cells are showing signs of accelerated growth, resulting in cell division and proliferation division, and the tumor is growing. So the whole issue is that the gene affects the receptors. What proves this is the second experiment we're seeing. They take very thin slices of this cursed cancer, the malignant one that doesn't respond to treatment, and they'll see what the cells look like. Here, we'll find the cell wall is thick, so thick that the cells look like circles. See how thick the wall is? Why is it thick? Because there's a cascade of growth signals constantly bombarding it, coming from its receptors. There's activity in the wall itself, a source of tremendous electrical signals commanding growth. Whereas this is the terrible cancerous tumor but in tumors that aren't, the wall is barely visible. And it's all due to these receptors. What's happening in the cell receptors? So he said, well, let's think of a good idea. Let's create antibodies for these receptors. And these antibodies will paralyze the receptors. Like this idea. Since the issue is with the receptors, Let's make antibodies to paralyze the receptor's activity. So he went to a large company that makes antibodies and vaccines and things like that, and asked them to prepare a vaccine for the receptors specific to this particular aggressive cancer. It took 2 years. The company spent 2 years preparing these antibodies that target the receptors in the cancer cell wall. And now all that's left is to test it. Test what? He put ads in the newspaper looking for volunteers, and at the same time he started testing it on mice. These mice, he first induced cancer in them. He took cells from breast cancer and injected them into the mouse, and then he'll test what? See, the poor mouse is getting a tumor as a result of what? He'll test the antibodies this company made on it, and at the same time, see other mice that didn't get the antibodies and see the difference. And he found a big difference indeed. This mouse that didn't get antibodies, see how the tumor multiplied? Now we'll see the mouse that got the antibodies that the company made. See, it's clear there's a big difference, that the antibodies really shrunk and reduced it, while the mouse that didn't take it was full of tumors. He said this is a very encouraging experiment to try on a human, and he announced it in the newspapers and asked for volunteers. So a volunteer came, which is this woman, Madam McKinley, who says she's been trying for 2 years with all methods to stop the growth of breast cancer she has without hope, and the chemicals aren't doing anything, and the radiation isn't doing anything. The only remaining hope is this discovery. She's getting on the plane and going to Los Angeles. And on the plane, she's writing her memoirs. She says the cancer is spreading in my breast, and there's no hope from any radiation or chemotherapy. The only thing left is for this discovery that Dr. Suleiman discovered to be the cure. These are the last words she writes while on the plane. And she imagines and hopes that this treatment will really work. She and her husband are going to the hospital for the scheduled interview. And she also describes her feelings that she only has this glimmer of hope left. And that this is the last thing left, that she's tried every treatment and nothing works. Dr. Suleiman, from the expected meeting, will first examine the eye to make sure that the cancer hasn't reached the brain and hasn't made secondaries in the brain. He's telling her he'll give her a double dose to get a clear result, hoping, hey, for the best. Okay. He'll examine the eye closely. See the back of the eye. No spread to the brain yet, which is good. They've brought the magic. KI and it's the messy horror. One is glucose, the other antibodies. They'll inject directly into the blood 5 times from the required dose. He wants to know for sure. Eliminate all doubt. He hopes there are no side effects from these antibodies, no impact on normal cells. Antibody means targeted at a specific cell, a specific target, not just any cell. It's against something specific, like receptors of a certain cell. But sometimes these things have side effects. But everyone's optimistic. As we said, he gave a double dose 5 times the dose. Then he'll do something else important, give her a radioactive injection. This injection will let us track where the antibodies go using a gamma camera. The radiation attaches to the antibodies, making them radioactive. Then he can track where they went via gamma camera imaging. Here's the radioactive injection. Then they'll wait a week for the antibodies to travel and settle on their target. Then they'll use the gamma camera to see what happened. Here's the gamma camera after a week. They'll see where the antibodies went and great. The antibodies all gathered in the breast area, specifically in the cancerous tumor. This means the antibody went where it was supposed to the receptors. Mama. So it must have blocked the receptors. We'll see what effect this has. These receptors are supposed to grab onto it and remove it, stopping the flood of growth and division signal so that the tumor stops growing. Responding to chemo. The doctor says we'll need several treatments. She said, did. After a week, she felt intense itching in the breast area where the antibodies gathered, meaning there's a battle or an effect happening, showing as an urge to itch. She also said the tumor started responding, which is great news. Yuskiwakwer. Another researcher, Banamji Batsky Dean, had aggressive breast cancer and was unsure how to treat it. Because she researches genes, she was incredibly motivated to find a solution, as she was also about to die. So she had a personal drive to discover something. What did Batsky discover? She found some cancerous tumors spread quickly and others and others don't. She said there must be a gene behind it. She researched the fast-spreading tumors and discovered a gene called NM23. If this gene is active, it prevents spreading. If it's inactive, the cancer spreads. So a gene controls cancer spread. This gene, NM23, prevents cancer spread when active. Surprisingly, she found this gene in a type of fungus called slime mold. This experiment will show the difference between cancer cells that spread and those that don't, hence the different color. The dark yellow, brown ones are the ones that spread. It means the NM23 gene isn't active. So the image shows a color difference. It really means this gene impacts cancer spread. His puss. And like we said, she found it in slime mold. It controls the mold's spread. We're still on the first breast image. This is slime mold. This slime mold, spreads fast. When the weather changes and dries, it stops spreading. It stops. The NM23 gene controls this, yes. Meaning the same gene controlling cancer, spread controls this mold's spread. The question is, can we treat fast-spreading cancers or with NM23? That's the researcher's natural thought. She wants to find a way to control cancer spread, which will kill her, who's at the body. But the problem is, NM23 can be present inactive and have no UN effect. Or it can activate. The exciting thing about genes is it's a code in the DNA. My dear Nicholas Pay, its command is suspended. It may or may not activate. There can be a cancer gene, but it's dormant. It's inactive and has no effect. What does this mean? It means the gene isn't fate's maker, as geneticists first thought. When they found genes, they thought they held fate's keys. But that's wrong. The gene isn't fate's maker because it can be a cancer gene, dormant and inactive. Then suddenly it activates. So the gene itself doesn't make fate. Is the gene just a cause? And behind every cause, there's a causer. This causer wills or doesn't will, meaning he's in charge of everything, of man, his DNA, and his genes. This Almighty Lord is the one who permits or doesn't permit any action to occur. God's hidden will is what activates the gene's switch so it works. God erases what he wills and confirms, and with him is the mother of the book. Meaning, there's the small booklet inside our cells, which is the DNA, which is a copy of what we said is a copy of the preserved tablet in the DNA. But God has the big book, which is the mother of the book. God erases what he wills and confirms, and with him is the mother of the book. Meaning that God Almighty erases what he wills and confirms from what is recorded in the decrees of this book. And this means the absolute freedom of the divine will. God Almighty does what he wills. Genes can never control God's will. Genes are just causes. God can stop these causes, and God can push them to work. Genes are not fate makers, but genes are just suggestions. Just a draft, just a blueprint, as they say. But the final decision is in God's hand. He erases what he wills and confirms that there is no will above God's will, even if it's the initial plan that God deposited in us. And the matter, my brothers, will depend on our behavior, how we will live a life full of alcohol, smoking, and surrender to vulgar desires and lusts. A hidden hand will press a switch. Cervical cancer, breast cancer, AIDS, syphilis, gonorrhea, lung cancer. All this can happen, and the ordeal begins. Meaning God gave us freedom and a margin of permission to choose the right life and the wrong life as we want to choose. We've been given leeway and freedom to choose the right path or the wrong one. Thus, we're each responsible for ourselves. We can destroy ourselves or save ourselves. Each of us now plays a role in our destiny. The final decision rests with God. He'll open the door of salvation to those who repent and seek forgiveness, and close the door of mercy to the stubborn and persistent. So our freedom exists all the time, and God's will is always active, and we play a role in our health and sickness. The book of genes says this through the tale of amazing genes that activate or don't. This is from the wonders of the genetic code and the wonders of the DNA strand. A miniature version of the preserved tablet within the nucleus of our cells. Until the next episode, God willing, and peace, mercy, and blessings of God be upon you.